Kevin Davis here at Exploring Next Generation Sequencing, the CHI conference in Providence, Rhode Island, and I'm joined by Jonathan Rothberg. Hey, Jonathan.
Thank you for having me today.
Jonathan opened the meeting. He is the founder of too many companies to mention, but some include Curagen, 454 Life Sciences, Raindance, and Iron Torrent Systems. We'll try and touch on all of those. So, Jonathan, you, of course, made your first mark in the genomics field, launching Curagen, and I guess that chapter kind of closed in a way with the company was recently sold. Looking back on Curagen, pride, missed opportunities. What's your sense?
First, pride. We were one of the first companies really to focus on mining the human genome as opposed to sequencing the human genome, and really made me think a lot about how do we get biological information into the digital domain. As you know, at the height of my pride was 1999. Curagen was worth $5 billion. We had 500 employees. And my son was born, rushed to the newborn intensive care, and really shift a lot of my focus, instead of understanding disease in general, on personal medicine, and that led to the development of 454, where I then put my efforts.
Right. So 454, it took 6 years from that sort of eureka moment where you saw a picture on a magazine cover of an Intel Pentium chip, and just explain briefly what did that image do in terms of thinking how you should reinvent DNA sequencing?
Basically, when I saw that Pentium, I realized that our approaches had been wrong. Whether it was Curagen, which was doing the Henry Ford assembly line and using the recently released browsers and the internet to tie automation with informatics, and I realized what we really needed to do was shrink the thing, really take the monolithic idea, and it was the first time, one, we sequenced on a single chip, massively parallel, which became the standard, and second, that we wouldn't use bacteria for cloning, we would use limited dilution. So really two eurekas there, but it's one thing to have a eureka, and it's another thing to get a great team to turn it into product, and that's what we did at 454. And we had a lot of firsts. It was pretty good. First to launch, first to do a human with Jim Watson on a chip, and first human publicly released the Neanderthal genome, the honeybee transplantation, so it was fantastic. And as an evangelist of a technology, I like combining that. I like inventing, but I like evangelizing. So it was a wonderful journey.
You spoke in your talk about the honeybee, the colony collapse disorder, and of course you were co-authors with Ian Lipkin at Columbia on a paper incriminating the Israeli acute paralysis virus, and you mentioned some perhaps unpublished data, but there's further corroboration now of that of the role of that virus in the colony collapse disorder, correct?
Absolutely, but I want to make clear, this wasn't 454's science. 454 invented the technology, and I had gone to Ian and said, hey, let's sequence something from the atmosphere or the environment or blood, and it was really collaborators of Ian that came and said, hey, let's do the honeybee. And so our contribution was the technology, and then at 454 under my leadership, we did the sequencing. But the science and the framing of the honeybee question was theirs. We did the work and the tools. So that was exciting, and it had an impact. The second question, though, is it's one thing to have a correlation. Hey, we found a new virus that caused the collapse, or in another case, we found a virus that was killing transplant patients. It's another thing to close the loop, and again, as an evangelist, we leave it up to our collaborators to close the loop. And so Ian has informed me that people have taken the virus and infected, and according to, I guess, Koch's hypothesis, if you have an agent and then you can finish the circle and infect it, you've done it. In the case of the transplant patients, they just had more immunochemistry, and others, no one actually infected people to prove that did it.
Another, I think, great thrill for you personally was getting Jim Watson's sequence and working with him. Tell me about that. How has he reacted to having his genome? My sense is he sort of perhaps was expecting more than anybody was really able to deliver, just given the state of the science at that time.
I love the question about Jim Watson. When I first approached Jim Watson, he was very generous, very gracious, gave me the genome. Later he said he gave it to me because he didn't think Jonathan would be able to sequence it. But actually Friday he came up to me, actually Thursday, let me correct myself. Thursday,
last week at the Cold Spring Harbor meeting.
Came up to me and he said, Jonathan, thanks for making me relevant again. So it was funny because he's in the news all the time again. But Jim Watson was the right person. He gave us the basis of heredity, and even more than the basis of heredity, he gave us the mechanism for all the sequencing technologies. Any sequencing technology you look at is based on the fact that this helix, when ripped apart, sets up a mirror for a twin of it. And for the basis of copying it. And every sequencing technology now uses that to sequence. So he gave us really the basis of heredity and the basis of sequencing, if you will. You have one template, you make another template.
Now, you've stepped away from 454, obviously, with the sale of the company that you founded to Roche a few years ago. I'm curious, how do you handicap the sequencing field these days? As a somewhat impartial observer, we'll get to your latest venture in just a second, but how do you see, is it developing the way you thought it was? Are there new technologies that are surprising you in the speed with which they're coming online? Is the dominance of one company surprising you?
I think what's interesting to me is it's made it clear that sequencing is evolving more in parallel with computing than I would have ever imagined. And in computing, we had a few centers that had mainframes and could support a mainframe. And in sequencing, we had these large centers, which were the only ones that really had the capability to participate in the first consensus human genome project. Then 454 and then others which followed that all use a monolithic idea, and all of them use sequencing by dilution, really have created in my mind what I call the mini computers. And when I say mini computer, I don't mean this. I mean these VAXs that then there were 400 universities or so or more that could use. And so what's really interesting to me is this evolution in clusters to a mainframe period, which was the Sanger labs, Sanger sequencing and the genome centers, to this mini computer, which is really the next 200 to 400 labs. And so I think it's really solidified my thought that they've made mainframes, and it's really time to step back and say, does it end? I'm sorry, they've made mainframes, then they made mini computers, and does it end with mini computers? And most of us actually know this story, and the answer is no, it doesn't. It ends when you can much more efficiently get that chemical information into the digital realm. And so I would tell you that we're somewhere around the late 1970s, to take the computer analogy. So it's just started.
Right. You've launched another company called Raindance. Just tell us a little bit about how things are going there.
Raindance is really a focus on the underlying technology that we used for 454. And at 454, we were single-minded. We said, we want to use this technology, these droplets, and putting these droplets on a single substrate to do one thing and one thing well, and sequence. And if you want to get to market and have an impact, you have to focus. At Raindance, we stepped back and said, wait a minute, droplets work, things on a substrate work, let's make something more general. But as we discussed, general things don't sell, specific solutions sell. So Raindance evolved to being able to do a tremendous amount of things on a chip, from immunocytochemistry to the evolution of enzymes. But the application, its killer app, is a DNA-based application where we're pulling out amplicons or exons from the genome. And I actually think this really matches and fits well into this paradigm of this mainframe period, mini computer period, because the mini computers, the next gens, don't have enough power to sequence 10 to 30,000 people, which we really need to do to understand disease. However, using ideas like Raindance, and other people have clever ideas to pull out exons, you can sequence that 1%, making you 100 times more efficient, and just focus on the exons during this period. So use the mainframes in the best way we can before we have this next great leap.
And in your talk this morning, Jonathan, you gave a tantalizing brief glimpse as to the sort of the focus of your next venture, Ion Torrent Systems, which has sort of been in stealth mode for a good couple of years now. Just recap what you told the audience. What can you tell us about the niche that Ion Torrent is hoping to fill?
Well, with my experience at Curigen, trying to span this chemical digital divide, 454 and Raindance, and even looking at the other players like Affymetrix with bringing in photolithography, I was able to step back again. Literally, my son really did start it again. He said, Jonathan, what do you want to do? A genome sequence? I want to read someone's mind.
Does your son call you Jonathan?
Full disclosure, Noah doesn't, but my daughter Alana does. When she wants to get me attention, she says Jonathan. Noah does say Dad. So he said, I want to read minds, and I stepped back and said, wait a minute, what has my career been about? My career has been about this general information. Turns out neurological information is only one. And you do want some kind of sensor that you could put into somebody's brain and directly read these things out. And so it allowed me to rethink, and also rethink what happened to each of these companies. Why did we fall or others fall? And if you look at Affymetrix, they got in trouble because they spent $200 million on wafers that they had problems manufacturing. And those slipped, allowed other people to come into the industry. I'm not going to point out the faults of companies I was involved in, but there's this whole supply chain. You can't just make a new technology, you have to distribute it, it has to be cheap, you have to supply for it. And really it came to the revelation that you cannot solve these problems by spending $1 million or spending $100 million of investor money, or even spending 200 or $1 billion. They're more expensive. And if you look at the semiconductor industry, and it was motivated somewhat by trying to get to the moon, a $20 billion project. And you look at the semiconductor industry and you calculate how much they put into making these iPhones possible, making it possible that the camera in here costs $3. It's $1 trillion. So you realize that the only way to go is to leverage this $1 trillion. Because if I raise $200 million, I'm going to burn it up on a plant. Or I'm not going to have the efficiency. You see these people scanning and it takes weeks, or they're scanning and they have to run around with hard drives, or they're scanning, they have to go fast enough, and they have technologies don't scale. So I see this in both the next gen technologies, these mini computers, as well as I see people now making what I would call the supercomputers. That they want to do, genomes so fast, but I think underneath they have this flaw, which is it doesn't cost $100 or $200 million to solve these problems. These are $1 trillion problems. And that might sound insane, but we actually have a workbench and a tool set from a $1 trillion problem, which is the semiconductor industry. So I'm pretty convinced on the end, the answer is going to be one sequence, one transistor, just like it is with memory and just like it's becoming with imaging.
And are you looking for partners to help you develop this technology? In other words, is this sort of an enabling technology that will have many applications? I'm just curious if commercial groups should be contacting you to sort of share ideas.
Good question. Basically, we're interested in having a direct interface between that chemical world and the digital world. And obviously, as I mentioned, platforms don't sell anything. You have to have killer applications. And so we're thinking about those things.
Well, Jonathan, good luck with those, and we hope we see you back here at one of these meetings in the not-too-distant future to reveal the full secrets of ion torrent systems. I'm Kevin Davis here in Providence at Exploring Next Gen Sequencing with Jonathan Rothberg. Thanks for watching.
Thank you.